Hello my dear students, I hope you all have been doing well. Here in this lecture, this lecture is the continuation of my previous lecture that was about protein structure. So in that lecture we would be discussing about the secondary structure of protein. So now we will be continue from the previous lecture. So we would be discussing in our previous lecture about the handedness of the beta strand. So the beta strands are having the handedness. What does it means? It means that they can be right or left handed. So this one we can see here that figure 2a, this is the right handed form of the beta strand, and the figure 2b is the left handed form. And now we are going to discuss about another protein secondary structure, that is the Betatrend. So what are the Betatrend? Actually the Betatrend are the most common form of the trend, and Betatrend actually a type of a non-regular secondary structure, and then the trend is composed or consists on 4 residues of amino acid. So what is the amino acid residue? Actually, you know, the amino acid is a monomer that is having an alpha carbon and joined with 4 different other groups. One of them is carbonyl group, one carboxylic group, one is the amino group, one is the hydrogen atom, and one is the R side chain. So when amino acid start making peptide bond and amino acid start joining with each other via the peptide bond. So how this peptide bond form? After the removal of a water molecule from the 2 amino acid. How this water molecule remove from the 2 amino acid? One amino acid carboxyl group remove hydroxyl group, and other amino acid amine group remove the hydrogen atom. So and then make a water molecule, and subsequently a peptide bond form between 2 amino acid. So here I am explaining you what is residue. So residue is that portion of amino acid that is remain after removal of a hydrogen from amine group and after removal of a hydroxyl group from carboxylic group of the amino acid. So here we can see that what is a Betatrend? Betatrend is a type of a non-regular secondary structure that is having 4 amino acid residue, and it is found in the protein, and due to the these amino acid 4 amino acid in the Betatrend, so this protein can change the direction. So here we can see that we are having 2 beta plated sheets, and here is a trend. So due to this presence of this trend, Betatrend, this polypeptide chain can easily change its direction from. So what amino acid that could be formed in the Betatrend are the cysteine, serine, and then proline. Most important amino acid that form in the Betatrend is the proline. So we have already discussed about the proline amino acid, that it is having aliphatic side chain that is cyclic in nature and having amino group rather than the amino group. Due to the presence of this amino group, proline can easily be bent. So what type of amino acid would be found in the residue are one of the most important type is proline, and other than serine and other amino acid could be found in this Betatrend. And what happened in the Betatrend, carboxylic group of one amino acid, oxygen atom of the carboxylic group of one amino acid would form the hydrogen bond with the amino group of another amino would be form hydrogen bond with the hydrogen atom of the amide group of another amino acid that could be found across from the amino acid. So and then this bond would be formed with the amino acid that would be present 3 amino acid apart. So here we can see that every time the bond is forming with the amino acid that would be present on the third position. So this is the one, second and third. So here this is how this Betatrend is important for the protein and the polypeptide chain. Why it is important? Because due to the having Betatrend protein beta strand or protein polypeptide chain can easily rotate in another direction. Now we would be discussing here another type of protein secondary structure, that is a coil. And whatever the protein that are present in the cell, almost 40 to 50% protein are present in the coil form. And what is the coil? Actually coil is a protein secondary structure that could be anything except the alpha helix, beta sheet and Betatrend. So in the protein, whatever you are having, whatever structure you are having, whatever secondary structure you are having except the beta sheet, alpha helix or Betatrend, it would be the coil. So and one important type of coil that we are discussing here is a leucine zipper. And now we would be studying about what is the loop. Loop is also a protein secondary structure, and we can see here a picture. First of all, we would be discussing that what is a loop. Loop is a irregular structure which connect two secondary structure element in the protein. So loop is a irregular and it can be found to join two secondary structure, means it could be formed to in the middle of a Betatrend or two Betatrend, or it can be found to be joined one alpha helix with the Betatrend, or it could be found to join one beta plated sheet with the alpha helix. So the structure, the secondary structure that is present in middle of two regular secondary structure of protein is a Betatrend that is irregular in nature. And Betatrend varies in length and its 3D configuration, and it is not regular, and the amino acid of loop. So amino acid of loops are polar in nature mostly. So what is the protein loop? Loop is a secondary structure and of protein, and it is irregular in nature itself, but it is found to join two regular secondary structure of within a protein, and it is basically composed on a polar amino acid, and due to this reason it is mainly found on the surface of the protein. And why, question is this here, that why this loop and trun are important for protein. So actually loops and trun are important for protein because they make able to the other secondary structure in the protein to join with each other. So as we have already discussed that sometime loop is found to connect one of the alpha helix with other of the beta strand, or sometime it found to connect two Betatrend, and sometime it may be found to connect two alpha helices. So due to this connection, protein can exhibit various conformation, and protein can able to fold itself. And then a loop, due to the nature of the amino acid that are found in the loop, these amino acid are mostly hydrophilic in nature. So loop always found on the surface of the protein, and loop contain a maximum or more than or less than 5 residues. So everything except the regular secondary structure of protein that is composed of 5 irregular 5 amino acid, more or less 5 amino acid, and irregular in nature and found to connect to regular secondary structure are called as loop. So loop is an irregular secondary structure of protein. However, alpha helices, beta sheet, beta strand are regular secondary structure. And due to the now this was about the importance of loop, and now we would be talking about the why the Betatrend are important for the protein. So Betatrend are important for the protein because the it helps to secondary structure or a secondary structure to change its direction. So for example, if the trunk connect to beta strand, so due to the Betatrend, this beta strand can be converted from the parallel means beta strand to anti-parallel, means due to having trunk in the loop, a beta strand or beta sheet can be means change its direction. So the trunk, why is trunk is important for the protein? The trunk is important for the protein because if protein polypeptide chain is having a Betatrend, so due to this Betatrend, protein polypeptide chain can change its direction. So now we would be talking about another secondary structure, and it is the bulge. So what is a bulge? A bulge is a non-repetitive structure that can only occur by itself in the coil region. So always this secondary structure, that is bulge, will always be found in the coil region, and it found mostly in the coils. Means what was the coil? Coil would be the when the protein polypeptide chain will fold itself. So it was the coil. But when the non-repetitive structure and what is the bulge? Bulge is a non-repetitive structure that will be found in the coil, and it is means irregular in nature, means not regular. However, beta sheets and alpha helix are regular. So it is a regular structure, but it is visualized as an irregularity in antiparallel beta sheets. So what is a bulge? Bulge is a regular, well-defined regular structure that found in the coil, and in the coil it is a non-repetitive structure, and however if it will be present in the beta sheet, so it will be found as a irregular. Beta bulge is a regular itself, but it will, when find in the beta sheets, it is considered as the beta sheet is, means it will be considered as the irregularity in the beta sheets. So this was whole about the different secondary structure of protein. So what secondary structure we have studied till now? We have studied loop, bulge, and coil, Betatrend, and alpha helix, beta strand, and beta sheets. So all these are the secondary structure of the protein. And what are the primary structure of protein? Primary structure of protein are the sequence of amino acid in the space, or in the biological, means you can say in the space, the sequence of amino acid is a primary structure. And then if secondary structure are repetitive structure, reoccurring or repetitive, means pattern that protein polypeptide chain exhibit. So they can be beta bulge, they can be loop, they can be trans, they can be beta sheets, they can be alpha helix, and they can be beta strand. Now here we are seeing that this chart is showing you about the probability of occurrence of a different type of amino acid in different secondary structures. So here we can see that how we read this chart, we can see that the probability probability of alanine amino acid would the probability of presence of amino acid alanine in the alpha helix is 1.29 times, and in beta sheets 0.9 times, and in the reverse turn 0.78 times. So here we can see that what amino acid would be found in least means in least what amino acid is having least probability to be found in the alpha helix. This one is the glycine. So, and here we can see that here we can see that what the amino acid that found the higher probability in the alpha helix would be what would be? It would be spartate. So this is how we read this graph, and then we are talking about the Ramachandran plots, and actually what plot it is and why we use it, this plot actually, and how we found that if we are having a protein, how we can find that what type of secondary structure, what is the probability of the protein that it is having what type of secondary structure. So we can easily found, if we are having an unknown protein, so we can easily found that what type of secondary structure and what proportion of what secondary structure it is having with the help of this Ramachandran plot. So what is this Ramachandran plot? There was a scientist who was Ramachandran. He actually identifies different angles in the protein polypeptide chain that were the Ramachandran angles, and we have already studied in lecture number 2 what are the Ramachandran angles. These are the pi and Pasi angles. And what are the pi and Pasi angles? The pi and Pasi angles are actually those angles that are formed between C alpha C and NC alpha atom. These atoms are these angle are the angle that are formed between the C alpha C bond or NC alpha bond. And what are these bonds? These bond are those bonds that are adjacent to a peptide bond, and that these bonds are not rigid, these are flexible, and we say these bond as a pi and Pasi bond. And due to the presence of this bond adjacent to the peptide bond, the polypeptide chain can easily rotate, because the peptide bond is itself is a rigid in nature and cannot rotate. However, the bond that are present adjacent to the peptide bond, that are pi and Pasi bond, can be easily rotate in the space, and due to the rotation of this pi and Pasi bond, protein polypeptide chain can exhibit various conformation in the space. And then how we find that what conformation is protein is having? By the calculation of this pi and Pasi bond. And how we do this? We do this by using how we identify what conformation of protein is having by the Ramachandran plot. So this Ramachandran plot, we plot the pi angle on the x-axis and the Pasi angle on the y-axis, and by doing so, if the protein is having the alpha helices, so it will be accommodate in this region, and if the protein is having beta sheets, so it will be will be accommodate in this region where the angle would be plus 180. So this is how we can find out the different conformation or different secondary structure of protein, what type of structure secondary structure protein is having. So this is the other this is this Ramachandran plot of a protein, and here we can see that this is the this was the region of the alpha helices, and this was the region of the beta sheets. So all those the beta sheets, if the protein is having the beta sheets, so it will be appear from here, and all those the protein that all those the alpha helices that protein would be having, it will appear in this region. So Now, except the primary structure, secondary structure, tertiary structure and quaternary structure, protein is also having one more structure, that is super secondary structure. And the super secondary structure of protein actually connect, we can say that it is a making a pipeline between the secondary structure and the tertiary structure of protein. So first protein exhibit the primary structure, that is the sequence of amino acid in this space, and this then secondary structure, that is the repeating of the polypeptide chain in this space or the repeating unit. And then we will be having super secondary structure. And what are the super secondary structure? Actually these are the joining of different secondary structure together, or the connection of different type of secondary structure together. And after this super secondary structure, proteins fold itself in the three-dimensional way, and it will be the tertiary structure. And after that we would be studying the what is the quaternary structure. So here we are studying about the what is the secondary structure of protein. The secondary actually here we are studying about the what is super secondary structure of protein. So actually the super secondary structure of protein is that structure that would be formed after the connection of two and more secondary structure of protein or secondary structure motifs. So and what are the secondary structure? There are different type. What are the super secondary structure? There are different ways, or by that protein can exhibit super secondary structure, and we say that these means ways as a motifs. So a protein can have the alpha helix motif, and then as a super secondary structure, and beta motifs and the alpha beta motifs. So why these are motifs? Means when two or more only alpha helixes join together, so it will be alpha motifs, alpha helix motifs. So in the alpha helix motifs, only alpha helix would be present. Actually, what is the helix? Helix is a secondary structure of a protein, and when two or more helix would join with each other via loop or turn, so it will be a alpha helix motifs. And what is a beta motifs? Beta motifs would be formed after joining of two and more beta plated sheets or beta strands. So here we are seeing that if two beta plated sheet would be joined with a loop, so it will form a beta hairpin loop, and it is a motif. This is a super secondary structure protein. If a motif would be formed by by joining of four different beta plated sheet via a loop, so it will be another type of beta motif, or one another type of beta motif. It is a beta barrel. So what is a beta barrel? Actually in this beta barrel, it is a type of super secondary structure. And in this beta barrel, how this beta barrel form? It is formed by the by a joining of multiple beta sheets via the loops. So that is how it can be possible that we are having the motifs that are having both alpha sheets and beta alpha helix and beta sheets. So in this scenario, we will say this type of a super secondary structure as alpha beta motifs. So what is the alpha beta motifs? Alpha beta motifs are actually the combination of different type of motifs, that is alpha helix, different types of secondary structure of protein. They may be alpha helix and beta plated sheet. So this was about the super secondary structure of proteins. The super secondary structure of protein are actually the motifs. They may be same. They may be formed between same secondary structure, and they may be formed between different secondary structure of protein. So now we would be studying about the what is the tertiary structure of protein. So actually the tertiary structure of a protein is a three-dimensional structure of a protein, in that a polypeptide chain that is having more than one secondary structure folds itself in three-dimensional way. So here we can see that this this polypeptide chain is having beta plated sheet, and it is having the alpha helices, and it fold itself it fold on itself in a three-dimensional way. So actually the 3D structure or third degree structure or three-dimensional structure of a protein is that structure in that a polypeptide chain fold itself in three-dimensional way. So and in this what happened in this type of a protein, all those amino acids that would be hydrophobic in nature would be buried inside this three-dimensional structure and inside the polypeptide chain. However, whatever amino acid they would be hydrophilic in nature and positively or negatively charged would be present on the surface of a protein, because of the having charges they will be attract with water, so they will come over the surface of the protein. And one more thing that we have to discuss here, that the disulfide bond will be helpful in this will be helpful to form this 3D structure of a protein. So we have already studied in our first lecture, that was the about amino acid. What amino acid form the disulfide bond? That was the cysteine amino acid. So cysteine amino acid, when oxidized, it convert into dimeric amino acid, that is the cysteine amino acid. So actually if what happened when a protein polypeptide chain that would be having a beta sheet or alpha helix, it will be fold itself. So what happened that amino acid cysteine would get oxidized, and then would maybe oxidized, and then if it will encounter the environment that can oxidize it, and then the amino acid make the disulfide bridge in between the cysteine amino acid. So due to this disulfide bridge, protein different secondary structures or different parts of the polypeptide chain get attached together. So one more important thing that we have to study here, that what type of a secondary structure means, what is the domain and what is the fold. So this fold and domain would be included as in the secondary structure of a protein. And what is the domain? Domain is a region of the protein polypeptide chain that is self-stabilizing, that can self-fold on itself. So if we are having protein that is 2000 amino acid long, but if initial 200 or 500 amino acid have capability to fold itself and exhibit 3D structure, however the other amino acid would remain straight. So the portion of that polypeptide chain that would be having the ability to fold itself separately will be called as our domain. So our domain is a region of a polypeptide chain that can be self-stabilized. And what is the stability of a protein? Actually, when protein folds itself, so it will be stable. Because why it would be stable? Because its peptide bonds would be protected from the biological environment, and it will be less hydrolyzed. And when the polypeptide chain would be in extended conformation, unfolded conformation. So the peptide bond would be more exposed to the biological environment and could be easily hydrolyzed, and protein could be degraded or amino acid can be released from the polypeptide chain. So what is a domain? Domain is that portion of a protein that can be self-stabilized or that can be means exhibiting the 3D structure of a protein. And then what is a fold? A fold is a isolates of discrete structural similarity within which structure share some level of sequence similarity. So all this fold and protein are the part of the 3D structure of a protein. And what is a 3D structure? A 3D structure of a protein is that structure in that protein polypeptide chain can exhibit three-dimensional means configuration or conformation, three-dimensional means shape in the cell. So here we are taking an example of 3D structure of a protein, that is a myoglobin, and it is this structure could be identified via X-ray crystallography during 1958. So the 3D structure of a protein could be seen like this. So here we can see that a protein polypeptide chain that self means fold itself in a 3D way. So now one more important thing that we have to study here, that we are having different techniques by which we can find out the 3D structure of a protein. So what are these techniques? These are the X-ray. One of the most important technique is the X-ray crystallography. And by the X-ray crystallography we can determine the protein structure, and the we can also this technique cannot give us the information about the conformational flexibility. However, we are having another technique, that is the nuclear magnetic resonance, and this technique can provide us information about the conformational changes. What are the changes protein are exhibiting, and what is the structure protein are having. However, this technique provide low resolution structure of protein. Now we are having another technique for the to find out the 3D structure of protein, is the cryogenic electron microscopy, and this technique provide us the quaternary structure and the tertiary, both quaternary and tertiary structure of the protein. And then we are having another technique, that is dual polarization interferometry, and this technique provide us information about the surface capture or complementarity in the protein. So this was all about the 3D structure of protein, how 3D structure of protein could be formed, and what are the 3D structure of a protein, and what are those techniques that could be used to find out the 3D structure of a protein. Now we would be studying about the quaternary structure of a protein. So what is the quaternary structure of a protein? Actually, the quaternary structure of a protein is the organization of the subunit of a protein with the other subunit. So first thing is that here, what is a subunit? Actually, a subunit of a protein is a single protein molecule that can assemble with other protein molecule to form a protein complex. So here we are seeing an example of a protein, and this is actually example of a blood protein, that is hemoglobin, and it is having 4 different subunits. So we can see here the different 4 subunits are depicting here in different colors. So each color is showing a separate subunit. So here we can see that different subunit are assembled together. So when the different independent subunit of a protein join with each other via different forces, weak or non-covalent forces like hydrophobic and electrostatic interaction, so they make a structure that we say as a quaternary structure of a protein. So what is a quaternary structure of a protein? Actually, the quaternary structure of a protein is the assembling of a independent subunit of a protein via different non-covalent weak interactions. So till here we have completed all our different structures of a protein. So to summarize again, what we have studied till now, we have studied till now that what are the amino acid. Actually, amino acid are a monomeric molecule that are having an alpha carbon atom that is joined with the amino group and a carboxylic group and a hydrogen atom and a side chain. And different amino acid having the different side chain, and there are 20 amino acids that are existing in nature, and all these amino acids are different with each other due to the having differences in the side chain of the amino acid. And when these amino acids would join with each other via the peptide bond, so they make a polypeptide chain, and this sequence of amino acid in the space or a polypeptide chain is the primary structure of a protein. And what is the secondary structure of a protein? When this polypeptide chain will fold itself, or we can say when this polypeptide chain will exhibit the repeated sequence or repeated arrangement in itself, so we will say this is the secondary structure of a protein. For example, the alpha helix. So in the alpha helix, how this polypeptide chain is having reoccurring arrangement, it is in the form of a coiling. So what is the tertiary structure of a protein? When the polypeptide chain that is having both alpha helix and beta sheets fold on itself in 3D way, so it will be tertiary structure of a protein. And what is a quaternary structure of a protein? When this independent subunit, when two or more independent subunit of a protein join with each other via different non-covalent forces, so they form a complex molecule of a protein that we say as quaternary structure of a protein, that would be having multiple subunit of protein molecule. So this was a whole about the protein structure. Now in my coming lecture we would be studying about how protein perform different function in our body, and what is the biological and the pharmaceutical importance of the protein. So these are some references you can go through with this, and then for more reading I would suggest you the book Larringer. And thank you very much for your kind attention, and Allah Hafiz.